So, share screen. Should work. Let's see. Okay, it is Monday, October 12th. I thought we were going to have Zoom meetings at 6:30, but obviously nothing's gone right this semester. So I'll wait about 5 minutes or so before I get started to see who else is going to join us. Right. Okay, so today I want to go over chapter 4, at least part A. Part B, if more people join, we'll see what's going on. Hopefully everyone has figured out how to get into mastering to do the lab exercises. And if you can't get in from lab for somehow jump in lecture, they should have been copied. So do them in lecture and I can manually input the grades into your lab. I'm pretty sure this morning I redid the test, so it should be open. It was supposed to open Friday. I set it up at round 2 to open, and apparently it didn't. So there's that. All right. So I know everyone's a little frustrated, so please be patient. This obviously is not going as well as it could be or should be. So between the Zoom account and YouTube, we should be able to get this all up and underway. So chapter 4 will start unit 2, all right? And we have, I think, 3 or 4 more tests. I'm not quite sure. But remember, I dropped the lowest grade, all right? And your question should literally be very similar to what is in your lab. So whether you're in 151 with me or someone else, just realize that I'll copy the exercises to the lecture, so there's no reason why everyone shouldn't be doing well. I realize the lab exercises are going to take you 2, 3, or 4 hours to do. Please read the chapter beforehand. I guess we will kind of speed up the lectures so that it's covered here. And then if you have any questions with Zoom, I can answer them to some degree. And you guys should be in good shape. So please realize we're online. So there's going to be a lot of what am I trying to think of? Self-actualization. You're going to have to actually do some effort on your own to do this. I realize it's not the best situation, but it is what it is. We're in New York, and we have we have our governor, and he is setting precedents. So let's just try to be safe out there. So let's see what's going on. It's about 6:36. I'll wait another minute or so and see who's going to join. Like I said, I realize it's a holiday, but last week I got called into a meeting for the state boards, which is coming up in a week from today for me. So I will try to do the Zoom meeting from Albany. Yeah, either Sunday, Monday, Tuesday. So I'll do the meeting from my hotel room in Albany. So we'll see. All right, so I want to go over some tissues and some histology. Unfortunately, you guys aren't in lab or in lectures, so we can't look at individual slides and go over form follows function with you guys, but we'll do the best we can. So I'll just start, I guess. So individual body cells are specialized. So remember, form follows function. We're going to go over all different types of cells: squamous, columnar, cuboidal, stratified, unstratified. So please realize that each shape and layer is a function. Some of them are going to have cilia, some are going to have microvilli, some are going to have goblet cells that secrete mucin, and then when it comes into the fluid on the apical surface, it turns into mucus, and this is going to line pretty much every external portion of your epithelium, which is literally facing the outside of your body. So it's going to have a very protective function. And if we're talking about the digestive tract, please is going to have some absorption properties to absorb some of those nutrients. We're not quite there yet, but we realize that all mucous membranes have IgA, which is immunoglobulin A, which gives us some protection against outside invaders or things that we potentially don't want that could be theoretically pathogenic. So tissues are groups of cells, similar in structure that perform common or related functions. So these tissues, the cells are going to be all exactly the same, but they're going to be similar in shape. We wouldn't obviously have skeletal muscle cells or cardiac muscle cells somewhere where they didn't belong, or they didn't contribute to the functioning of the tissue. And then tissues are going to come together and produce organs, and then organs are going to come together to form organ systems, as we went over, and the organ systems are going to hopefully, with homeostasis, contribute to homeostasis of the body and be able to perform what they're designed to form. All right. So histology is the study of tissues, under slides or whatever. If we're looking at some kind of aspirated tissue or looking at some kind of biopsy, we want to be able to figure out what that tissue is, and if it is metaplasia or some kind of cancerous tissue, we want to be able to see if we can at least see the cell type of origin, just figure out, where this potentially came from, and if it spread through contact or metastate or metaplasia or whatever, we could kind of ascertain where it came from. All right, so there's 4 basic types of tissues. Epithelium is generally, like I said, always going to be on an apical surface. If it's the integumentary system, it's going to be outside of the body. It could also be inside the body, but generally they're going to, the apical surface is going to face the outside external environment. Connective tissue, we're going to learn, about different types of connective tissue, and these are for support. It could be reticular, it could be dense reticular, it could be tendons, ligaments, could be part of your shoulder, a capsule, whether it's in your knee or your shoulder, the femur, talk about muscle tissue now, whether that's skeletal, myocardium, which is heart, or smooth muscle, which is going to really control your, anything that's kind of a lumen or a tube is going to control peristalsis or movement in one direction, or, could be talking about the uterus, anything that's going to cause a motion in a certain way. And nervous tissue, we'll get a little bit into that. We'll talk more about that when we get to the end of the course, but nervous tissue is going to be sending messages to and from the brain center, the homunculus to be exact, which really is mapped out part of your brain that's going to control the impulses, so you know where it comes from. Then you send out an efferent impulse for an appropriate reaction to whatever the afferent signal is sending towards the brain. All right, so I'm going to move myself out of the way so I can read this. So nervous tissue, start here. Internal communication. All right, so brain, spinal cord, nerves. And just remember, the brain and spinal cord is the central nervous system. These are really not regenerative per se. There is some research showing that they can reproduce, but usually nervous tissue can't. The supporting cells, the glial cell, astrocytes, and dendritic cells, they can reproduce, but they're supportive cells. All right, and then our nerves, whether we're talking about afferent cells that are sending impulses to the brain for interpretation, and then efferent, it's going to send those signals back down the spinal cord for an appropriate response. Hopefully, if there's nothing affecting that function. And muscle tissue contracts to cause movement. So whether it's muscle cells, which are attached to skeletal cells through the periosteum, to through tendons, to through lever arms to control motion. Cardiac muscle, we're going to see coming up, they have intercalated discs, which are going to be able to tell each cell to communicate with the one next to it. So the heart can contract in a very specific way. So the atrium and ventricles contract when they're supposed to, so we have an efficient pump to pump that blood through the body. And then smooth muscle is going to be part of the walls of your hollow organs. So whether that is your digestive system, your ureters, reproductive system, so that they can move in a specific way. All right, epithelial tissue is probably the most prolific. It's going to reproduce the most, and it's probably the most numerous per surface area on the body. All right, so it forms boundaries between different environments. So whether that's the external environment, the internal environment, or different environments within your body, different body cavities, it's there to protect you from invaders or outside chemicals or sources or pathogens. It secretes. So whether it's going to be secreting through the endocrine glands, exocrine glands, or it could be secreting, like I mentioned, mucin, which turns into mucus, absorb. So if we're talking about the digestive tract, it's going to absorb certain substances, and in the uter, good grief, the nephrons and the kidneys or the digestive tract, it's going to be absorbed. And filtration, once again, in the kidney environment, the urinary tract environment, it's going to be filtering some things in and out of the system. Lining of the digestive organs and other hollow organs, glands, whether it's a pancreas secreting bicarbonate or amylase or lipase, something that's going to help you in the breakdown of things that you've consumed through the chyme in the stomach, or after you swallow that bolus, your food goes into the stomach for mechanical motion to give it more surface area, and then we're going to go into the duodenum, and that's attached to the pancreas, going to secrete some digestive enzymes for you. And the skin surface, you're probably all familiar with it, the epidermis. So that's going to secrete certain acids that has a salty environment to inhibit the majority of bacteria that can't live in that kind of environment. And then we'll get into connective tissue, which might be a little harder for everyone to get a grip on because it's a little more complicated, but the connective tissue is anything for support. So always remember if it's support. So protects, binds other tissues together. Now right here in the chart, it mentions bones, tendons. Bones support your structure, support movement. Tendons really connect the muscles to the bones. Ligaments will connect your bones to your bones. The fat and other soft padding for protection. And it really holds things in place, kind of like bubble wrap. And here it does not mention blood, but blood is one of the connective tissues that a lot of people forget. But remember, your blood is very supportive. And once again, it's the only way that things can get from anywhere in your body to anywhere in your body. All right. So microscope of human tissue. So if we were in lab, unfortunately not, we could give you slides and then could show you certain things with a pointer. And at school we have Lycra microscopes with cameras. We can project that and then capture the image and talk about it. But unfortunately, at the present time, we can't. But viewing under a microscope, we have a fixed tissue is preserved with a solvent. So we're going to do very, very thin slices. Now, whether in chapter 1, we talked about sagittal, transverse, oblique, or whatever. We can do very, very, very microscopic thin slices. Then we're going to stain them to contrast the components so we can actually view them through a light source and then project that onto your retina, and you can figure out what that tissue is, hopefully. So they'd be sectioned into slices thin enough to transmit light and electrons, and like I mentioned, they could be sagittal, transverse, oblique, mid-sagittal, whatever. We could come up with any term we wanted depending on, conditioning upon, what we sliced it. So if we were slicing the kidney nephrons, we want to slice them more of transversely, but they're moving in all different directions. So depending on where we slice it, that slice could be sagittal, transverse, oblique, or whatever. Really contingent upon how the slice came from and what portion of that they took it from. That's kind of an abstract image that you, over time, you just kind of get acclimated to that. So stain, like I said, we stain it to enhance contrast, although artifacts or distortions distract from what the sample looks like in living tissue. So when you're looking at a slide, you might see something black or something that just doesn't make any sense. Realize that can be an artifact. It could be an occlusion body, it could be part of the stain. So there are things on the slide that will show up as artifacts, and we really don't know exactly what it is. We can take a guess, but we don't know really ever what exactly it is. So light microscope uses colored dyes. So whether it's eosinophil or methylene blue, or if we're talking about bacteria, we're using a gram stain, Gram's iodine, or safranin, or different dyes depending on what contrast we want, or depending on whether we're looking at gram negative or gram positive, and the layer of the cell or bacterium. So electron microscope uses heavy metal coatings, and that can magnify things incredibly. You get a lot of detail using that. And sometimes we can bounce light off of that and ping it in a certain way. We can really get a 3D image of what we're looking at. All right, so here's a comparison. On the left, we have a light microscope. On the right, we have electron microscope. And that literally right here is a microvilli. I'm sorry, that's cilia. Sorry, in your respiratory tract. And that really is designed to move things like the wave in a certain direction. So generally, if that were the respiratory tract, we want to move that up towards to the upper respiratory tract so it can be expelled or swallowed, and then we can deal with it with some hydrochloric acid in the stomach. Right. All right. So epithelial tissue, or epithelium, depending on how they phrase it for you, is a sheet of cells that covers body surfaces or cavities. So this is really going to, in general, it's going to be the apical surface of that is going to be towards the outside of the body. All right. The basal surface is really, think of it as a basement or basement membrane. It's really going to be in contact with the blood supply or nutrients. All right. So there's two main forms: covering or lining epithelia on the external or internal surface. Example would be your skin. So the apical surface is on the outside. The basal surface is still cutaneous tissue. It's going to be more towards the basement membrane or the capillary bed of the blood supply. And glandular epithelium is secretory tissue in glands. So if it's glandular, realize your glands are going to be secreting something, whether it's saliva, it could be a mammary gland excreting milk in some sort. It could be a helican gland. It could be a pancreatic gland excreting some kind of digestive enzyme or whatever. And we'll get into that as we get into each section, whether it's digestive system or whatever. We'll get into that over and over again. So the main functions are protection, absorption, if we're talking about urinary system or digestive system, we're really going to absorb things. Filtration, so that could also be the urinary system, or filtration can be literally your alveolar sacs in your lung line. It can do that. Excretion, could be excreting, like if it's your skin, it can be secreting some kind of acid. Mucin gland secretion, or sensory reception. If we're talking about the afferent dendritic tissues that you can sense touch, pressure, proscenium corpuscles, or other sensory receptive parts of your afferent um, nervous system. All right, so epithelial tissue has five distinguishing characteristics. So it has polarity, so polar, or it has two ends. So we're going to talk about apical or a basal surface, has specialized contacts. We have something called contact inhibition between cells, which kind of tells the cells that if there's contact inhibition, so there's enough cells, it tells it not to regenerate, or if the apical surface keeps getting removed, the cells realize that they have to regenerate continuously. And please realize anything that has to regenerate continuously is going to have a lot of nuclei, because remember, the nucleus is the control center that's going to send out the DNA information to reproduce what it needs, proteins or whatever it has to make. All right, supported by connective tissue, and we'll get into that towards the end. So all these are going to have supportive tissue, whether it's adipose tissue, reticular, alveolar tissue, or whatever. It's going to have something to bind it together and protect it, whether we're talking about fibroblasts or other things that are going to have a supportive nature to keep that tissue alive and well. Avascular but innervated. So whenever you see avascular, A always means not. So if it's not vascularized, so some tissues like cartilage at the corner of your eye are avascular, all right? Which means there's no direct blood supply, but they are nourished by tissue. So if it is cartilage, they're nourished by synovial fluid, and through movement, we can move that fluid back and forth to give it nutrition. Or if it's a corner of the eye, your tears, all right, can give it nutrition. And then regeneration. So usually epithelial tissues have a lot of wear and tear, or they break down, so they have to be regenerated quite a bit, contingent upon what the environment's giving it, or what's going on with it. So polarity, cells have polarity. They have a top and bottom, all right? The apical surface is the upper free side exposed to surface or cavity. So if we're talking about your digestive tract or your respiratory tract, it's going to be that inside of that lumen, which is a hollow tube. Or if it's your skin, I mean, just look down at your arm or hand. So that external environment is the apical surface exposed to the outside. Right? Most apical surfaces are smooth, but some have specialized finger-like projections called microvilli. So the microvilli, there'll be a picture coming up. Those are going to look like cilia, but much smaller. They're going to increase the surface area of your digestive tract 1,000-fold. So when we talk about digestion, we can get into some of the digestive disorders like irritable bowel syndrome or celiac spree, or some of these diseases where the microvilli are constantly broken down or attacked by antigen-antibody complexes. And if they can't regenerate fast enough, you can have a lot of digestive issues because you're getting rid of really a lot of your surface area. All right, so there's not a lot of surface area for the absorption of the nutrients to go to the hepatic portal vein and go to the liver for processing. The basilar surface, the lower attached side, faces inward towards the body. So this goes in and hitting the basement membrane. And below that basement membrane is the capillary system. All right, so if we were talking about skin cancer or a basal cell carcinoma or something that was cancerous, as long as we can keep it above the basement membrane, it doesn't get into the capillary bed, it will not spread. But the minute it breaches that basilar surface and gets into the capillary bed, it can get dispersed through the body through the bloodstream, and now we have some kind of metaplasia. There's some kind of potentially spreading cancer cells. All right. So attached to the basilar lamina, an adhesive sheet that holds basal surface of the epithelial cells to underlying cells. We want that to be attached to something. So like if we're talking about your skin, or if we're talking about your digestive tract, as things move along or things brush against your skin, we don't want that skin coming detached. So we want something to cement it to. We'll talk about the connective tissue there as support. All right. right? And both surfaces differ in structure and function. And we'll see that coming up here. All right, so that's epithelium. Epi always means on. This is your apical surface, borders and open space. This is on the outside of the body. All right, so if you look, these are long columns. This is columnar. All right, so columnar has a lot of space in it. These could be a brush border. If you look, these very small, pretty tiny, like hair-like structures. So this could be this could be microvilli. This could be something in the ureters or something that's going to have, it's going to need something for increased surface area. Now the basal or basilar surface is next to the underlying connective tissue. So apical surface on the outside, basilar or basement is going to be the basal membrane. So these are going to be literally attached to this connective tissue down here. It's going to be there for adhesion. So it doesn't, you know, if we were thinking this was urine or food coming through here, we don't want these being torn off. We want them attached down here, right? So specialized contacts. Epithelial tissue needs to fit closely together. We don't want, I'm gonna go back for a second. So we don't want food or urine or whatever to seep into here. We want this to be pretty much fluid and airtight, right? So these have, they form continuous sheets, right? Specialized contact points bind adjacent epithelial cells together. So lateral contacts include, all right, so we're gonna have contacts all in through here, and desmosomes or things like that. They're gonna be like rivets holding these things together so they don't break apart, right? Tight junctions or desmosomes, right? All epithelial sheets are supported by connective tissue. So we have reticular lamina. This is very deep to the bottom, consists of a network of collagen fibers. So, you know, you may have heard of collagen or people get collagen injections. It's very, very durable material. So it's really gonna anchor things down. The basement membrane is made up of basal or reticular lamina. It reinforces the epithelial sheets, those sheets of cells all together. All right, restricts stretching and tearing of that, and it really defines the epithelial border. So we don't want that really, we don't want that breached whenever possible. All right. So as I mentioned, cancellous epithelial cells are not contained by the basement membrane boundary like other cells. They penetrate the boundary and invade underlying tissues, resulting in cancer. All right. So I'll go back here real quick. Sorry. So theoretically, we say if we had some kind of cancerous cell here and it were able to get its way through here and then get it under the basement membrane and get into the blood supply, we have a serious issue going on, right? Okay, so avascular but non-innervated. So remember, avascular is going to not have direct blood supply through the basement membrane, but it's innervated. So it's innervated for pain or pressure, so you can actually feel that. No blood vessels are found in the epithelial tissue. Most must be nourished by diffusion from other connective tissue. So, like I said, the cornea is avascular. A lot of the cartilage is avascular. So we have to, by pressure, moving those fluids back and forth, like squeezing a sponge or pushing things in, it's moved by diffusion. And if you remember the term diffusion, it's going to go from higher concentration to lower concentration. You guys remember that? Epithelia are supported by nerve fibers. All right, so they're supported by nerve fibers for pressure or pain. So, you know, everyone knows or has heard of bed sores or your cornea. You could scratch that, all right, if you sleep in your contacts or whatever. It's avascular, but you can tell if you get something in your eye, it hurts a lot. All right, so it is innervated, but it's not, doesn't have blood supply. Right. Regeneration. Epithelial cells have high regenerative capacities. And remember, these are exposed to the external environment and constant friction or things going by it. So they're going to slough off continuously. Stimulated by loss of apical basal polarity and broken lateral contact. So as these things abrade and there's no contact with them, they know, you know, there's an issue. So they're going to regenerate from the basement up, basal membrane up. They're going to regenerate themselves. And if it's epithelial tissue in the digestive tract or uterus, or, think about your esophagus. You're constantly swallowing things. It's going to slough that off. So it's constantly regenerating. Cells are exposed to friction, some hostile substances. So if it is your esophagus, whether it's alcohol or, whatever you're eating, if it's very, very hot or spicy or chemicals, it's going to really start eroding those tissues, results in damage. So they must be replaced. If they're not, then we have ulceration and we have all kinds of health problems. So we have to make sure that the repair process is on par with the breakdown. Yeah, so we have to repair as it's broken down. If it doesn't repair as quickly as it's broken down, then we have some degenerative changes and we have some potential pathological condition going on. Requires adequate nutrients and cell division. So we have to make sure we can innervate that with blood supply so it gets nutrients, it can run ATP. We have to be able to provide cell divisions. We have to be able to go through that mitotic division that we talked about. All right. So all epithelial tissue have two names. So remember it. Your first name is going to always be the number of cells. So whether it's one layer, two layer, multi-layer, it's always going to be simple, stratified. Simple is usually one layer. Stratified means it's in layers. All right. And then the second name is going to be the shape. So if it's squamous, they're really flat. If it's columnar, they're like cubes. I'm sorry, if it's cuboidal, it's cubes. If it's columnar, it is columns. So just think of how much area is inside that cytoplasm, and that'll kind of give you an idea of what's going on, because remember, form follows function. All right. So squamous are flattened scale-like. Generally, if they're simple squamous, they're going to be for diffusion or like alveolar or your parts of your kidneys. They're going to be for things moving back and forth very, very quickly. Or if they're stratified squamous, generally there's layer upon layer upon layer. They're really there for protection, all right, to make sure that things don't breach them and get into the basement membrane. All right, cuboidal are box-like cubes, and columnar is their tall column. So just realize with these cuboidal or columnar, there's going to be more area inside that cell. So if they're area inside the cell, it's not for protection. So think, well, is it going to be for absorption or excretion? Is it a big storage area or is it a small storage area? So does it need to hold a little bit or excrete a little bit, or are they going to be big cells for a lot of production of something? All right. If stratified epithelia, shape can vary in each layer. So cell is named according to the shape of the apical layer. And we're going to see coming up, you could have squamous the top and then cuboidal beneath it, or you could have squamous the top and then columnar underneath it. And then we can get into pseudostratified columnar epithelium and things like that. But in general, you always look at the apical surface. That's how the name's going to come from. The most superficial or the apical surface, whatever is exposed to the external environment, that's how we name it. So right here, we can see this is the apical surface exposed to the external environment. This is the basement or basilar membrane. All right. This is simple stratified. All right. But you could say, well, Dr. Field, it looks like there's cuboidal cells on there. There are. You're absolutely right. But the top layer is very, very thin. You can call them tiles or, however you want to remember them. That's up to you. All right. So the nucleus is usually always magic mouse here. The nucleus is always in the middle. All right. So if these are normal cells or parenchyma, the nucleus should always be generally in the middle of that cell. All right. You're always going to find exceptions, but in general, they're always in the middle. We're always going to name it by the top layer. Okay. So there's squamous. So these could be tiles. This is cuboidal. All right, or cube. This is columnar. And just realize, this is not producing a whole lot. It's not doing a whole lot. It's just there as protection, at least on the apical surface. Cuboidal, these can be for excretion or absorption, but not, they're not doing a lot. They're doing some, but not a lot. But a cube or columnar, all right? So usually the nucleus in a columnar cell is always towards the basal surface. And just realize this is a lot of area in it. All right, so it's going to be there for something. It's going to have a very specific purpose. All right. So simple alveoli involved in absorption, secretion, and filtration purposes. All right. So simple squamous epithelium cells are flattened laterally and cytoplasm is very sparse. Function where rapid diffusion is a priority. So generally, like I said, the kidneys, the lungs for the alveolar diffuse that oxygen, carbon dioxide through. All right. Two very special simple squamous epithelium are based on their locations. So the lining of the lymphatic vessels, blood vessels, and heart. That's endothelium. Endo always means inside. So you're going to line some kind of a vessel. All right, lymphatic vessels we'll talk about. They run along with your nerves, arteries, and veins, and the heart. The endothelium, the inside lining of your heart, is endothelium. Right? Mesothelium comes from the middle layer during embryonic development. These are serous membranes in the ventral body. Can't remember, ventral is always posterior. And serous membranes. So serous membranes are going to produce serous fluid. They're going to produce fluid, and serous fluid generally is for lubrication. So think of it as if you had a Ziploc bag and you had some water in it. All right, you want a little fluid in there so you could move it back and forth. All right, so just realize you have these membranes surrounding anything that moves continuously. So whether it's the heart or lungs, they're constantly moving. You don't want any friction. All right, similar to oil in your engine, you want something there to absorb heat, so there's no friction heating and friction rubbing against those tissues. All right. So here's a picture of the squamous epithelium. And remember, this generally is for diffusion. And if you see, this is your alveolar, this pink space. This is where the alveolar of your lung, the distal end of your lung or your bronchi. This is where oxygen and carbon dioxide are going to diffuse. We want this very, very thin for diffusion. All right. So if you had pneumonia or something and you had a lot of fluid buildup in here, the oxygen and carbon dioxide wouldn't be able to diffuse back and forth very well. You'd have a problem perfusing the tissues with that. All right. So simple cuboidal epithelium, remember simple is going to mean one layer. Cuboidal is going to give us the shape, and endothelium tells us this is going to be facing the outside surface. So whether it is kidney or wherever, we'll see coming up, wherever it is. Single layer of cells involved in secretion or absorption. Forms walls of the smallest ducts or glands of many kidney tubules. So these are going to be actually ducts or glands. They're not going to be really there for diffusion. They're going to be excreting or absorbing something. Right? So whether they're absorbing glucose from the filtrate in the kidneys, or they're absorbing something along the way, or if they're ducts, if they're secreting something into a duct. A tubular duct as opposed to a tertiary duct. All right. So here's a single layer of cuboidal cells with large spherical nucleus. So I said there's always generally going to be in the middle of the cell. All right, here's your nucleus, always in the middle. There's your basement membrane. So they're attached to something. This in here, if you look at this space in here, that's the tubule. Right, that's the lumen of the inside opening. So this is, you have to picture this coming up out of the slide here in columns. So this could be a tubular. So realize this was sliced this way, all right? But this tube, these are glomeruli, or these are I'm sorry, this is the nephron, the tube of the kidney. All right, so this is going to be the lumen. So this is going to be absorbing or moving things back and forth. As the filtrate moves through the kidney, it's going to be absorbing things that we want to absorb. Anything we don't want, we're going to let pass through the tubule. We're going to excrete that through the distal proximal tubule into the urine. All right, so it becomes urine. After it becomes filtrate, turns into urine. Simple columnar epithelium. Remember, there's a one layer columnar. They're going to be very, very long, or longer than cuboidal. And in general, the nucleus is always towards the basilar surface of this. All right, and that, if it doesn't make sense, realize I want that nucleus towards the bottom, towards the basement membrane, just for protection. We don't want anything happen to the nucleus. Remember, it has all that genetic DNA in it. So some cells are microvilli. This is for surface area. And some have cilia, and that's for motion in a one-way direction. All right, some layers contain mucus-secreting goblet cells. All right, so your book says here, and the slide says these goblet cells secrete mucus. Technically, that's not correct. They secrete mucin. All right, and when mucin comes in contact with water, it turns into mucus. All right, so Mucinex, all right, that medication that helps you expel all that mucus is involved in that process. Ciliated cells move mucus found in the digestive tract. And that's microvilli for surface area for absorption of nutrients. Gallbladder, all right, ducts from some glands, bronchi, and the uterine tubes. All right, uterine tubes could be, cilia has that to move the egg down in the correct direction. All right, and there's an example of it. All right, so if you look here, these are long. This is simple because there's one layer. And remember, the nucleus is generally towards the basement membrane. All right, here, that's a goblet cell, a mucus cell, a mucus of goblet cell. And remember, realize these are really towards the apical surface. So here's a goblet cell. That's literally, we did the slide, or they did the slide, in the process of bursting that gland open, and it's secreting that mucus into the tract. And remember, if this is digestive tract or uterine tract or whatever, we want the mucus there. We want that as not slime, I guess slimy layer to help things move along. We want to give it a little bit of lubrication, right? Okay. Hold on 1 second. I'm gonna let somebody in. Right. Oh. Okay, I think she's in. Her video's off. All right, Katie, can you hear me?
Yes.
Oh, I hear something. Okay. All right, so pseudostratified, all right. Okay. All right, pseudo means false and stratified. So this is going to look like it's in layers, but it's really not. So if you look, some of the cells are not going to go from the basement membrane all the way to the apical surface. They're really going to be, some are going to be contained towards the apical surface. Some are going to be more contained towards the basilar surface. But when you look at it initially, it's going to look like they're layered. But look closely, because a lot of the cells won't go all the way from the basilar surface all the way to the apical surface. So just look at the difference of that. All right, cells vary in height and appear to be multi-layered and stratified, but the tissue is in fact single-layered, simple epithelium, and they're going to crowd each other out. So some are going to come to a point, and then there's going to be ones on top of it. So look closely, and you'll see their pseudo means fake. They're fakely stratified. All right, many of the cells are ciliated. Remember, if they're ciliated, that's going to be for motion in a certain direction. All right, so these are going to involve in secretion, particularly of mucus. So if they're secreting mucus, they're going to have goblet cells. They're going to secrete that mucin, and then it comes to the apical surface. It comes in contact with water. It's going to turn into mucus. All right. Located mostly in the upper respiratory tract, duct of large glands and tubulars in the testes. There's Sertoli cells that are important in producing and secreting semen. Right. So if you look, kind of here, I'll give you an example. So if you look, that one, if you looked at this closely, you'd say, oh, it looks like there's 2 layers. That's, definitely not simple cells, but it actually is. It's pseudostratified. So it looks like it's stratified, but literally there's only 1 cell here. This one's making a layer out of it. It goes all the way to the top, but this one, so if not all of them go from the basement membrane to the apical surface, there wouldn't be simple, right? All right, and there again, there's that goblet cell, and these are going to be more towards the apical surface because their job, here's one excreting it, is to literally build mucin. When they push them up, they're going to come in contact with the fluid out here, they're going to it's going to turn to mucus. We want that for some lubrication. We want to be able to move that that food through the digestive tract, right? Or if we're talking about the respiratory tract here, we want to be able to, that cilia, to move anything that these ciliated cells have gotten along. We want to move it up towards the throat so we can expel it or re-swallow it, right? So stratified epithelial tissue. So stratified, once again, remember this is more than one layer. So it could have 2, could have 10. It depends on the protective needs of that tissue or that area of the body. If it's under a lot of abrasion, a lot of chemical assaults, we want to have many, many layers. Involve 2 or more layers. New layers are generated from below. Remember, I said they come from the basement membrane towards the surface. All right. More durable than simple epithelium because protection is their major role. They're going to have many, many layers. We want, if the first one erodes off, we know that from the contact, non-contact with other cells, it's going to tell the cells below it, listen, you need to replace that layer. It's going to send out signals. It's going to regenerate that tissue. It regenerates contingent upon the damage of it, the layer above it. All right, stratified squamous epithelium. So stratified is going to be many layers. Squamous epithelium. We said before, squamous was those tile-like cells, very, very thin. You can think of them as tiles or pennies or dimes or whatever you want to think of them as. All right, so these are stratified in many, many layers, and that's going to be for protection. So as that top apical layer sheds off, the basement membrane moves those tissues up, all right, to regenerate that. Oops, what did I just do? Okay. All right, located in areas of high wear and tear, example of your skin. So the outside of your skin, whether you know it or not, it's completely dead. It's dead keratinized tissue, and as it goes from the basement membrane up, they become less and less alive, and the top surface of your skin is completely dead, all right? And that's what causes dust in your house or whatever as it wears off, all right? Keratinized cells found in skin. Non-keratinized cells are found in the moist lining. So if it's in your skin, they're keratinized protein, all right? Non-keratinized cells are the ones found in moist linings, all right? So here's stratified squamous. So if you look, these cells are really, really thin, but if you look, there's many, 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 many layers. And then here's your basement membrane. All right, so these cells are going to regenerate from here. These are very, very big and alive. So as these wear off, it sends signals down here. These cells down here are going to start pushing the cells up here. And as they push them up, they become less and less alive until they get to the surface here. They're completely dead, and they're just keratinized protein. They're just there for protection. Right. Right. So stratified cuboidal epithelium is very rare because remember we said that cuboidal is really going to be there for diffusion or absorption. So we really wouldn't want them layered because that would the layer below the top layer wouldn't have any surface area or any wouldn't have any surface area for diffusion or filtration, all right? But these are found in some sweat and mammillary glands, are typically only 2 cell layers thick. And that's in case the top layer breaks off, we already have the bottom layer ready to move up into place and take its place. All right. Stratified columnar epithelium, also very limited distribution in the body because once again, if they're columnar, they have all that space. They're literally there for storage or excretion. Right? Small amounts are found in the pharynx, the male urethra, and lining some glandular ducts. Usually occurs at transitional areas between other 2 types of epithelium. Only apical layer is columnar. All right, so the top layer is going to be columnar. Okay. All right, transitional epithelium. This is only found generally in the urinary system, and transitional. All right, so that transitional means it's able to change shape. This is for excuse me, anything that has to stretch or give. And if you look at the cells, they're going to be very, very curved. All right, and that's like if you're talking about your bladder, bladder is empty, it's very, very small. But if it fills, that tissue has to be able to expand or change shape. All right, So basal layer cells are cuboidal or columnar. Ability to change shape when stretched allows for increased flow of urine. In case of bladder, more storage space. Okay. So here's transitional epithelium. Now, if you look, these are very curvilinear, I guess you could call that. These are very, very curved, all right? And that's so it's able to expand if it needs to, all right? And literally, if this was the bladder here or the ureter here, if there's more fluid, these cells have to be able to push down and expand, okay? All right, so again, one or more cells that makes or secretes an aqueous fluid is called a secretion, are classified by site of product release. So endo, we usually always think of internal or like endothelium, all right, internally secreting like hormones. They're going to internally secrete, endocrine glands are going to secrete hormones into your blood supply, and they're going to find their target organ through receptors and have that response to whatever specific target. So if it was your thyroid gland, so thyroxine or whatever, is going to find that gland through very specific receptors, all right, and generally turn that gland on or off depending on what was needed. Exo means external, externally secreting, all right, so sweat. So your exocrine glands are going to, or halocrine or apocrine glands or whatever, are going to excrete sweat. Sometimes they secrete fats and oils, and that can contribute to bacterial breakdown and sweat and body odor and whatever. All right, relate relative number of cells forming the gland. So unicellular, meaning one. Goblet cells, and we saw pictures of one of them that go towards the apical surface. They are exocrine glands. They excrete that mucin, turns into mucus. Or if they're multicellular, we're going to have salivary glands or American glands or like mammillary glands are going to be multicellular, they're going to be excreting a lot of substance. So here is invaginated epithelial sheet. So look, these are contiguous. These are columnar cells. They're going to be excreting that to the apical surface right there. All right, here's an exocrine gland retaining the connective cells, which form a duct that transports secretions to the epithelium. So this is going to be this similar thing, but see this big area here? All right, that's going to have a lot. These are cells are going to be excreting to this duct, and then the duct is going to excrete this, whatever it is, to the apical surface or the external environment. And this will be excreting much more than this would be. All right, endocrine glands, these are going to be, if you look, endocrine was always in this side. These are endocrine glands. They're going to excrete this. And there's your blood supply. It's going to take this to whatever, and take it through the capillary system, your capillary system either in the near area or through your arterioles, your arteries, and secreting it around the body. And these hormones are going to, when they get to the certain area in the body where that receptive gland is, they're going to migrate out of the capillary bed and turn the receptor on to that organ. Okay. So endocrine glands are ductless. Secretions are not released into a duct, are released to the surrounding interstitial fluid, which is picked up by the circulatory system. And they secrete through exo, remember exo's out, exocytosis, hormones, messenger chemicals that travel through the lymph or blood to their specific target organs. Target organs respond in some characteristic way. You're going to turn it on or turn it off, right? Depending on homeostasis and what the brain center is telling, the hypothalamus is telling it to do, right? And we're going to the endocrine system, I think, ANP2, we'll get right into that, right? So exocrine glands, remember the exo means out. Secretions are released onto body surfaces such as skin or body cavities. More numerous than endocrine glands, right? Secrete products into ducts. These are mucus, sweat, oil, salivary glands. Right? They can be one cell or they can be multicellular, depending on how much they really need to secrete, contingent upon what their needs are. Unicellular, like we saw, mucus cells or goblet cells. So there's just one cell and they come to the apical surface, they excrete their mucin, it turns into mucus, all right. And then we see this, there's your goblet cell, right? And as it comes to the surface, we see here, it literally excretes that. And there's your, you know, nucleolus, right? There's your Golgi apparatus. Remember, these are preparing this. We put it into vesicles. We transport it into exocytosis. It's going to leave that cell. All right, hopefully this is tying some of the stuff that we talked about last unit, making a little bit more sense, or why do we need to know that? Well, here's an example of why. All right, so multicellular exocrine glands are composed of a duct and secretory unit, are usually surrounded by supportive connective tissue that supplies blood and nerve fibers to the glands. So the glands have to, you know, if they need blood to get oxygen to produce ATP, to do all this. So they're very, very active. They need some blood supply. They need nerve innervation, right, for function. Right. They're classified by either their structure or how do they secrete it, right? Simple, right? So you're saying simple, well, it should be very small, maybe one layered, right? Have unbranched ducts, but compound glands have branched ducts. So if it's simple, it's going to have one little duct. We saw an example of that with that simple tubular, right? Compound are going to have more than one gland, and usually they're kind of like a, I guess you could call it a T-shape. You know, it really depends, and there's normal variance in every area. So in a tubular gland, secretory cells form a duct, whereas in alveolar glands, secretory cells form sacs. So remember, they're forming sacs, all right? They're secreting a lot more because there's a more area, right? Tubular alveolar glands have both types. So they're just saying tubular, so they just kind of combine that name. So remember, there's always exceptions, and they just kind of combine that. It's more of a compound type gland, all right? So here's simple duct structure, all right? It's pretty simple, all right? You know, might have a little T structure. But if we get into the compound, just realize, imagine how much these are producing, all right? Surface area, and all this area is producing going out of the duct. So the duct, so these are going to secrete far larger quantities of whatever it's secreting. All right, so mode of secretion. Mericin glands, merocrine, most secrete products of exocytosis. So remember, that's leaving the cell, going to the cell wall, and then merging with the cell wall and excreting it in the environment. All right, sweat or pancreas. Remember, pancreas is going to secrete those digestive enzymes. Holocrine, accumulate products within, then rupture. All right, so holocrine glands literally, they kill themselves. All right, sebaceous oil glands, they rupture. They generally die, they have to be replenished. So halogen, they accumulate products within them, they get super, super big, then they rupture, sebaceous oil glands. So this is really what produces the oils and stuff that bacteria tend to feed on. And that really, not to be gross, but it really contributes to body odor. And this can be more prevalent in reproductive areas below your navel, right? So they can be more prevalent down there. Apocrine, APO, accumulate products within, but only apex structures where this type exists in humans is controversial. So maybe mammillary cells, but there's still no definitive proof on that. All right. So these are Mericin glands. All right. So they secrete their products through exocytosis out. All right. These are holocrine glands. They literally, these cells, these are going to excrete it. They're still alive. These literally get big and they just kill themselves. They explode. Well, explode, but they rupture. All right. All right, guys. I think that's enough for tonight. I know it's my fault. I'm kind of late doing this, but like I said, I will plan unless something else comes up. Like it called Albany or whatever, but we'll figure doing this on Mondays at 6:30. If someone needs to do it earlier, if earlier is better, let me know. I only had one person join tonight, so if another time works, let me know. I can joggle this around. All right, so please let me know. All right.